In 1993, I started my degree. I started an anatomical sciences degree at Cardiff University, and in the third year of that degree, I took on a research project which would make up my final year thesis and probably the bulk of my mark for the third year and the bulk of my mark for the degree. That research project was trying to answer the question, why can some frogs stick their tongues further out from their mouths than others? Is it something to do with the way the collagen fibres are arranged that support the muscle? So I spent a lot of time looking at tongues that I had dissected and prepared under a light microscope and under a scanning electron microscope. So getting around to look at the histology of the tongue is a fun throwback. It was that research project that made me realise I wanted to become a scientist, and I did a PhD, and then I ended up here somehow. So I've got 3 slides. Don't worry, they're not going to take too long to look at. The reason we've got 3 slides is because, well, we're looking at the tongue, sure, great place to look at skeletal muscle, we'll find some minor salivary glands, but the things we're really interested in are taste buds. We want to look at actual taste buds under the microscope. And the tongue has got a structure, I'll pinch this out of big head, in that we have papillae. So there are some filiform papillae over most of the tongue, and those are just rough shapes to the mucosa that let us grip food and move it around our mouths. And then we have these recesses around here, and then we have other papillae around here. So we have the fungiform papillae, we have the foliate papillae. Back here there's a V of valate or circumvallate papillae. What I'm saying is I can't look at all those in one section. So we've got different sections of the tongue so that we can look at all the different papillae, we can look at the different taste buds. All right, now this is going to be, I think, surprisingly pretty. Surprising for you, not for me. Busman's holiday for me, isn't it? Okay, let's have a look. Excellent, Masson's trichrome as well. So Masson's trichrome is, rather than just hematoxylin and eosin, where everything's a bit purple and a bit pink, here we've also got green showing connective tissue. So the tongue is largely skeletal muscle. We can choose to move our tongues, and we have, this is the lowest power. This is the 4 times objective, 10 times to my eyes, so 40 times magnification. And what we're looking at there are muscle fibres running in different directions, arranged into bundles. So it's those bundles we were looking at, collagen fibres wrapping around those bundles. That's why I needed to use a scanning electron microscope, so I could look at this in higher power than we can see with a light microscope. But the tongue, people think the tongue is a special muscle because it's only attached at one end, but that's not what's going on here. You've got extrinsic muscles that move the base of the tongue around, but these intrinsic muscles of the tongue, inside the tongue, are attached to the inside of the mucosa of the tongue. Look, that's what we can see there. So that's the other thing that we're interested in, is the mucosa. So instead of having skin, when we go inside the oral cavity, we've got a mucosa, very, very similar, a little bit different, and the muscle is actually attached to the connective tissue supporting the mucosa. So when the muscle contracts, it pulls on the inside of the tongue, changes the shape of the tongue, so the volume remains the same, but if you make it narrower, it gets longer, you make it fatter, it gets shorter. That's how you can stick your tongue out and move the tip of the tongue around. And what we're seeing on the top there, look at those beautiful spikes. Those are the filiform papillae. Now, the epithelium of the mucosa in the tongue is a stratified squamous epithelium. So it's a multi-layered epithelium of squamous cells, flat cells. Of course it's an epithelium. And this is because well, the tongue and the oral cavity sees a lot of wear and tear because food is passing through here, right? So there's a lot of mechanical abrasion, cells get lost, new cells get replaced from the lower layers. And generally this is non-keratinized. So we have a keratinized stratified squamous epithelium in our skin, which makes us waterproof. The keratin makes the cells waterproof. What we've got here is with the filiform papillae, with these spiky papillae, it is lightly keratinized, and that helps develop those spikes, and that gives us the friction that we need to move the food around inside our mouths. And you know, some animals like cats have got really, really rough tongues. This is what you're feeling when you feel the roughness of that animal's tongue, these filiform papillae. Didn't I see? Told you this was going to be unexpectedly pretty. Look at those shapes. Fantastic. I love it. My eyes are going funny, but there we go. Struggling to get a focus. So I'm struggling to get a focus, I think, because of the depth and the layers. So the green there is the connective tissue supporting the epithelium. The purple is the epithelium. And you can see, just like in the skin, how we have a basal layer. Those will be the progenitor cells producing new cells. As the top ones are lost, we've got those keratinized cells closest to the surface. Those are performing that mechanical function, and in between we've got cells on their way through that process to become keratinocytes. Oh, look, right there. That is a fungiform papillae. So we have a mix then. So throughout most of the tongue, this is the anterior 2/3 of the tongue where we find these papillae and these taste buds. Most of the tongue is covered in filiform papillae, and you can check that yourself by feeling the texture of your tongue. But we've also got lots of little lumps and bumps all over the tongue, and that's these fungiform papillae. Now, a fungiform papillae is fungus shaped. The chemicals in the food will collect in there or on the surface, and they will be presented to the taste bud, and that is what I'm seeing right on the surface there, right on the top. That is a taste bud. You see that little round thing, and there's a little aperture at the top, which is what connects it to the outside world. That's where the chemical reaction occurs. So here we have some specialized oral epithelial cells, and we have some cells that can generate an action potential when a chemical cue triggers that cell to produce the action potential. That runs to the brain, and we perceive that as taste, be it, you know, bitter, salty, sweet, sour, umami. So that was pretty good, wasn't it? Straight away. Can you see the difference? So those are the filiform papillae, and there's the fungiform papillae. And just looking at this section, we can see, well, I think so, we can see that there are far fewer fungiform papillae in this section, and very, very many filiform papillae. So we've talked about the mucosa, we've talked about the connective tissue that's supporting it, and as I rove around, you can see these blocks. Oh, we can see these blocks of muscle running in lots of different directions. So here then, there we go. That's all pretty lovely skeletal muscle. We can see some blood vessels. See the cells inside the blood vessels there. There'll be nerves in here as well. Yeah, there's nerves there. But the bit I should really highlight is this. So here we have skeletal muscle fibers running longitudinally along the length of the tongue and transversely, and we can see them running through there, and they've been cut in different directions. So the tongue is mostly skeletal muscle. Right, okay, so that slide lets us see filiform papillae and fungiform papillae, fortunately. The foliate papillae are in these ridges on the side here. I can't remember which of these is which. Let's have a look. I wanted to save the big ones for last, that's all. that's all. Oh yeah, here we go. So you can see here, now we've got a different stain. See, this isn't the mass and strichrome, this is your standard hematoxylin and eosin. And again, we can see, there we go. Those are salivary glands, minor salivary glands. But here we can see again, we can see all that skeletal muscle running through the tongue. And why has this suddenly gone so dirty? There it is, it was on the light source. Okay, so now you see this repeating pattern here. All right, so those are those ridges that we see on the side of the tongue. So these are what we call the foliate papillae. So these are quite large now. Look, I'm back on my 4 times objective lens, 10 times my eye, so 40 times magnification. And look at the size of the tongue. These are taking up a significant amount of space. We've got that gap, so it's like a moat. That's what the ridges are. You've got a gap in between each of these, and in there saliva collects and food starts to get dissolved. The chemicals get presented to the taste buds. Can you see the taste buds? There they are. So look, loads of taste buds in there. So that's what the foliate papillae look like. So the taste buds now are down in the base of the, this is 200 times magnification. The taste buds are down there, aren't they cool? So those are actual taste buds, though. That's the sensory apparatus that chemicals trigger cells to produce action potentials, and like I say, we perceive that as taste. So this is the foliate papillae on the side of the tongue. Look, see these taste buds down here, and here again. Now the other thing that we can see, so again, look, this is stratified. This is a stratified squamous epithelium, and it's not keratinized, but you can see those multiple layers of thin cells on the surface there. Oh yeah, the other thing that we can see is, so we could have seen better on the last one, but do you see how we have, if that's the mucosa there, it's like it interdigitates with the tissue underneath, so it gives a really strong grip. If this is like between the dermis and the epidermis in the skin, these get called just dermal papillae. But yeah, that's also a common feature then. The mucosa is interdigitated with the tissues underneath, so there's a strong grip. Now look, this here, this is glandular tissue, right? Pop that in the middle. So these are minor salivary glands within the tongue, so they're producing saliva. And we looked at the parotid gland last week, which is almost entirely serous. So you have, these are compound tubuloacinar glands, as in they're branched, they're tubular, they have secretory cells around that tube and they end with a bit of a curved cap, and they secrete into those ducts, and then they go into a single duct and leave. And there are serous glands and mucous glands. The serous glands are more watery and produce lots of proteins, enzymes and what have you to help with digestion. And the mucous glands produce mucin, which is a better lubricant. So these darker staining cells here will be serous secreting epithelial cells. Hopefully that looks familiar, hopefully that looks a bit glandular, hopefully it looks a bit like what we were looking at in the parotid gland. And then I saw some more lightly stained ones, I think. Oh yeah, so they were lower down. Yeah, look, see? So there we see serous minor salivary glands, and down here we're seeing mucous salivary glands, and we're getting a bit of that ductal structure that we looked at when we looked at the parotid gland. In that there's a duct there. Go and watch the parotid gland video if you want a bit more information about that, or I might describe it a bit better. But here again you can see cells arranged in circles around a tube. These are epithelial cells secreting into that tube, and they're mucus secreting, so they just, when you stain them like this, they just look a bit paler. Cool, I didn't think to look at these when we were talking about salivary glands. Right, so minor salivary glands. One more papilla to look at, then that must be this one. The circumvallate papillae, or the vallate papillae. These are so large that you can see them unaided very easily. So this is a slightly different stain again, isn't it? But look, we're seeing those salivary glands again. I don't know what stain this is. But look, okay, again I'm at my lowest power, so 40 times magnification, and this papilla is so big we can only get one of them in the section. So these are the papillae we see at the back of the tongue, so kind of separating the anterior 2/3 from the posterior 1/3 and forming this V shape here. So there's maybe 8 to 12 of these large papillae. They're kind of like the foliate, they're kind of like the fungiform papillae, like big fungi, but they're so much bigger. And like I say, they're arranged very specifically in this V. Fun fact, the depression at the point of the V here is the foramen cecum, and that's where your thyroid gland started to develop from in the embryo. Anyway, so these cells, same sort of pattern, big and special, but again they've got that moat, that ring around the outside, and again saliva collects in there, and of course now we see where some of the saliva is coming from because we've got all those salivary glands. Food gets broken down, chemicals get presented to the specialized specialized oral epithelium, and oh, we can just about see one taste bud in there. What about the other side? Oh, that's better, isn't it? We can see a whole bunch of taste buds down there. So look, they're not high up, they're low down, where you would expect the saliva and the chemicals to collect so that they can be presented to these taste buds. These look a little bit different, don't they? A little bit skinnier, a little bit pointier, but same principle, right? We've got that mucosa, but then we're kind of seeing these curved things. And depending on whether you get the planar section right or not, there will be like a central aperture exposing the cells in that taste bud to the fluid that collects in that moat around the vallate papillae. Oh, this is a real thick and wobbly folded section, isn't it? Because it's focusing a bit funny. But yeah, that's it for tongue, I think. Look, see, I have to go to the lowest power to get this whole thing in. I wonder if it's just H&E and faded or whether it's a different stain, but there we go. So again, lots of skeletal muscle. We can see those running in different directions. We're seeing serous salivary glands, minor salivary glands here, mucosa, connective tissue, and that circumvallate papilla. It is cool though, right? It's not just me. If I didn't think it was cool, I'd be in the wrong job, wouldn't I? But that is the histology of the tongue. Isn't it? It's just so good, isn't it, to look at it under the microscope, because we talk about taste buds, we talk about mucosa, we talk about all these things, but you can actually see it and you can see how it's arranged, and now you can better imagine, understand how the tongue works. So the tongue's functions are, you know, to move the food around to the teeth to help with chewing, to push the bolus of food to the back of the oral cavity for swallowing. So the filiform papillae is so important. Taste is so important to appetite and eating. One of the big problems that the head and neck cancer people tell me is that when their patients lose the sense of taste, you know, you worry about cancer patients losing weight, but when they lose the sense of taste, they don't want to eat. So they, you know, it's even harder to get them to eat. And taste, of course, is associated with smell as well, but those taste buds that we're seeing here, they're part of that safety mechanism that we have, figuring out whether what we're about to eat is safe or not, whether it's good for us or not. But those are the cells responsible for it. And then, of course, you see all these funny lumps and bumps on your tongue, and you're not entirely sure if they've always been there or if that's something new. That's what they are: the Filiform papillae, the Fungiform papillae, the Foliate papillae, and the Vallate papillae. So once you understand it and you know what they are, you know what they are. Okay. And yeah, if you're interested in skeletal muscles, it's a great place to look. Yeah. So what was the answer to the study? Were the collagen fibers arranged at a different angle in frogs that could throw their tongues out a long way to catch insects? No. Frogs that could throw their tongues out further, they cheated. They just had more tongue and they folded it up inside their mouths. The arrangement of connective tissue, the arrangement of the muscle was the same as we see in other mammals. They just had other mechanisms. A result is a result. We still answered the question. A good hypothesis means that either answer is a helpful answer, if you see what I mean. Yeah, funny to think that set me on my path. All right. Thank you for letting me look at the histology of the tongue. I won't see you next week. I'm taking a week off, but I'll see you in a couple of weeks.